This method can help reveal the differences between fungi and mammals at the molecular level. Such differences can be leveraged to identify new therapeutic approaches.
The main advantage of this technique is that it more efficiently modifies the genome of C. albicans than classic homologous recombination techniques.
To identify the guide RNA sequence, identify an NGG-PAM sequence close to where the stop codon will be inserted. Shown here are all PAM sequences found in the first 100 base pairs of TPK2, a cyclic AMP kinase catalytic subunit. Identify the forward guide primer 3 sequence. This sequence will be 20 bases directly upstream of an NGG-PAM site and will not contain more than 5 Ts in a row. Left-click on the base directly upstream of the NGG and drag the cursor 20 bases. Then, left-click on the Primer tab to add the primer. Now, identify the reverse guide primer 3 sequence, which will be the complement to the forward guide sequence. Left-click on the base directly upstream of the NGG and drag the cursor 20 bases. For each primer, left-click on the Primer tab to add the primer. Right-click the primer and select Copy Primer Data. Paste the sequences into a text editing program. Add overhang sequences to forward and reverse guide oligos to facilitate cloning. Add the nucleotide sequence ATTTG to the 5' end of the forward guide primer 3, and add G to the 3' end of the forward guide primer 3. Finally, add the nucleotide sequence AAAAC to the 5' end of the